Good morning, students. Let us begin with our today's topic. Our today's topic is mitosis. We had seen that mitosis and meiosis are the two ways of cell division. We had also studied that somatic cells and stem cells undergo mitosis, or the mode of cell division in these two types of cells is mitosis, whereas germ cells show the mode of cell division, that is meiosis. Today we are going to talk about mitosis. Let us first see the definition of mitosis. It is a type of cell division in which the chromosome number remains constant. Means if the parent cell has two sets of chromosomes, the new cells, which are also called as daughter cells, will also have two sets of chromosomes. So in mitosis, we find that the chromosome number remains constant in parent cell and the daughter cells. How many daughter cells are formed? Two. Two daughter cells, diploid cells are formed. So two daughter cells are formed from one parent cell and they are diploid cells. Diploid means which contains two sets of chromosomes. So one characteristic of mitosis is that chromosome number remains constant and two daughter cells are formed, and both these daughter cells are going to be diploid. That is, they will contain the same number of chromosomes as the parent cell and they will be diploid. That is, they will also have two sets of chromosomes as the parent cell. Now, mitosis is a type of cell division which is shown in asexual organisms. Sexual reproduction and asexual reproduction are the two modes of reproduction. Those organisms which show asexual mode of reproduction are going to divide by mitosis because there only one parent is involved, and since one parent is involved, we will find that the new cells that are formed or the daughter cells which are formed will have the same number of chromosome, or the chromosome number is going to be restored in the daughter cells as the parent cell. So in sexual mode of reproduction, two parents are involved. That is a totally different concept, and we will study it with respect to meiosis. Right now we are studying mitosis, and here we find that those organisms which reproduce by asexual mode of reproduction are going to show mitotic cell division. Only one parent is involved and the chromosome number remains the same in the parent and the daughter cells. What are the steps that are there in mitosis? In mitosis, there are two steps. The first step is called karyokinesis. Karyo means nucleus and kinesis means division. So the first step we find nuclear division taking place. In the second step, cytokinesis takes place. Cyto word is associated with cytoplasm. So we have cytoplasmic division. So there are two steps. There are four phases that are involved in mitosis. One is prophase, then metaphase, anaphase, and lastly telophase. Whereas here the very first one, that is interphase, is a preparatory phase. That is, the cell is preparing itself for mitosis or the cell division. So here you will find that the nuclear material goes on increasing and the cell prepares itself for cell division. So that is called as interphase. So what is mitosis? It's a process in which a single cell divides into two identical daughter cells, each having same number of chromosomes as the parent cell. The major purpose of mitosis is for growth and replacement of the worn out cells. So the major purpose of mitosis is the growth and development of the organism, and secondly to replace the worn out cells, because we have already seen that somatic cells, which are all body cells or the organs or the parts of the body, they are made out of somatic cells and they have got a definite lifespan and they need to be replaced, and so all somatic cells undergo mitosis so that the worn out dead cells can be replaced by the new one. So let us go on to the first phase, that is prophase. The first phase is prophase. Before prophase begins, there is a preparatory phase called as interphase. During this phase, the cell prepares itself for cell division. The cell grows in size. The DNA or the genetic material increases. We say that the DNA undergoes replication. Now, what is DNA replication? DNA replication refers to a biological process where we find that two identical replicas of DNA are formed from the original DNA molecule. I repeat, DNA replication is a biological process that is taking place inside the nucleus of the cell, and it is going to produce two identical copies of DNA from original one DNA molecule. So that is called DNA replication. So DNA replication is going to take place during the interphase. That is, the genetic material increases and the cell grows. Let us now see what changes happen in the very first phase, that is prophase. Condensation of thin thread-like chromosomes starts. So during interphase, you see that this genetic material which comprises of the chromosomes, they are thin and thread-like. But now during prophase, you will find that the condensation of these chromosomes starts. Now by condensation, we mean that they become short and thick. So here you can see that the chromosomes have become short and they have become thick. So that is called condensation. Sister chromatids appear. Sister chromatids, what do we mean by sister chromatids? They are two identical copies of the same chromosome formed by DNA replication attached to each other by centromere. So here you can see two identical copies of the chromosome. Here by color differentiation you have been shown that two identical copies of the same chromosome formed by DNA replication has been shown. So these are called as sister chromatids. So these two, you can say they are sister chromatids, or these two are sister chromatids. They are also called homologous chromosomes. Centrioles, which are present in the cytoplasm, duplicate and move towards poles. So here you can see these two towards the two extreme ends. So they are centrioles, and these centrioles are going to duplicate. That is, they are going to become double. So we have two centrioles now, and they start moving towards the two poles of the cell. And lastly, nuclear membrane and nucleolus disappears. So we, in the center of the cell, we have a nucleus which has got the genetic material, and the nucleus has got its own covering called nuclear membrane. So both nucleolus as well as the nuclear membrane starts disappearing. So these are some of the changes that we observe during prophase. One more thing, these sister chromatids which we see here, they are joined to one another at the centromere. So this is the centromere, and with the help of the centromere, the sister chromatids are attached to one another. Let's see what happens in the next phase, that is metaphase.